Go.
Hi, I'm Stephanie Street, senior biology student here at Winston-Salem State University, and I am going to help guide you through your sheep brain dissection today. Now, why are we using a sheep brain versus looking at a human brain? Well, one, a sheep brain is much easier to obtain. Two, it's a little bit smaller in size, so very easy to handle and manage while you're doing the dissection. And three, sheep brains are very similar in structure and function comparable to the human brain. So it's going to be very easy to identify the parts of the brain that is similar to be found in human brains. So let's get started. The first thing you need to do after you have rinsed off your brain and taken it out of the bag is identify the different directions of the brain. So if we're looking at the brain horizontally, we're going to be looking at a dorsal view, a ventral view, which basically means top for dorsal and bottom for ventral. We'll be also looking at a rostral or anterior view, which means front, or a caudal or posterior, which means back. So once you identify your different directions, let's start looking for the major regions of the brain. The first region is going to be the cerebral. It's going to be the largest region of the brain and the most easy to identify. The second is going to be your second largest region, the cerebellum. And third, the brain stem, which is going to be the most caudal portion of the brain, or the most ventral portion of the brain. So let's now identify the different lobes of the brain. First, you're going to make your brain vertical to you and identify your frontal lobe, where you're going to have cognitive and reasoning portion of your brain. Then you're going to have your parietal brain, which is going to be divided by a longitudinal fissure, which is going to separate your brain into left and right hemispheres. Secondly, let's turn your brain horizontal and find your temporal portion of your brain. And then you're going to find the occipital lobe. All of these major portions of the brains have different functions, which you will be identifying in your quiz. Now, let's start identifying parts of the anatomy of the brain. First and foremost, you want to hold the brain taut at the back and pull down so you can see the difference between the gyri, which are the folds of the brain, and the sulci, which are the grooves of the brain. Using your dissection kit, you'll be able to identify the folds versus the grooves. Now, when you look at your brain, you're going to also notice the different meninges. There are going to be three different types of meninges that you're going to be looking for. The first one is going to be the dura mater, which is the most superficial meninge of the brain. You'll note that you do not have that on this brain because it was removed when the brain was being removed out of the cranial skull. Second layer is going to be the arachnoid mater. That is going to be conducive to seeing all the small, spider-like capillary veins throughout the brain. And lastly, if you can pick it up, is the pia mater. This is going to be the most superficial meninge and the thinnest meninge on the brain. All of these protective coverings of the brain give the brain some protection from the skull. Moving on, let's start identifying your other structures that you'll need to know. So this first one was, we found the latitudinal fissure coming right down. If you'll note, if you can separate the brain slightly, you'll even see the connective fibers in between holding the two hemispheres together. Next, we're going to flip your brain to the dorsal view, and you're going to start identifying your olfactory bulbs. Well, it helps with your vision. Your optic nerve, right, octave chasm, and your octave nerves will be on either end of the octave chasm. What you'll find is that the nerves were cut once again when the brain was being removed out of the cranial skull. Then you'll be going and identifying your pituitary gland, which is right in this particular area. Then the rest of the brainstem that you're seeing on the ventral view will be showing the pons and the medulla oblongata.